In recent years, Neuralink, a brain-computer interface (BCI) company founded by Elon Musk in 2016, has garnered widespread attention in the tech world. The company focuses on developing implantable devices that enable interaction between the human brain and machines. This technology, known as a brain-computer interface, involves implanting tiny electrodes in the brain to facilitate interaction between computers and brain cells using electrical currents. It offers unprecedented therapeutic hope for areas such as motor disorders, neurodevelopmental disorders, and mental illnesses. Neuralink's BCI technology is invasive, requiring craniotomy surgery to implant electrodes near the cerebral cortex to directly collect high-quality brain signals. Despite the risks associated with surgery, biocompatibility issues, and long-term stability concerns, Neuralink has made significant progress in animal and human trials. It has reportedly completed the first human implantation surgery, with the recipient recovering well and already able to move a mouse cursor using brain signals. Neuralink's next-generation product, BlindSight, is particularly noteworthy. This product aims to restore vision to blind individuals, even allowing those born blind to experience the wonders of the world for the first time. It has reportedly received breakthrough device designation from the U.S. Food and Drug Administration, FDA, which means the device can receive earlier support from the FDA, accelerating its research and development and market approval process. If BlindSight can be successfully applied clinically, it will greatly improve the quality of life for blind people and provide them with unprecedented visual experiences. Beyond medical applications, what else can BCIs be used for? Uploading and downloading memories and knowledge? Today, let's discuss a different application of BCIs. In fact, BCI technology is not new, and its concept and exploration have a long history. As early as the 1870s, a pioneer in this field, British scientist Richard Cotton, successfully recorded electrical activity in animal brains outside the cerebral cortex using advanced experimental methods, laying the groundwork for subsequent BCI technology research. Moving forward to 1924, German Dr. Hans Berger took a historic step by placing electrodes directly on his son's scalp. This bold move not only demonstrated the fearless spirit of scientific exploration, but also achieved the first direct recording of human brain electrical activity, opening up a new path for the development of BCI technology. However, these recordings were all conducted externally on the scalp, capturing only the minute electrical signals leaked out by neurons, known as neural leakage, with extremely low accuracy. But soon, brain scientists invented new techniques capable of precisely recording the activity of individual neurons. Of course, this method had a small drawback: it required opening the brain. Specifically, a thin, insulated metal needle, with only the tip exposed, was used to drill a hole in the skull, insert the needle, and connect wires, thus creating the simplest form of a brain-computer interface. However, due to the destructive nature of the metal needle, this method could only detect the electrical currents outside neurons. Later, scientists replaced the metal needle with a finer glass pipette filled with potassium chloride as an electrolyte to conduct electricity, causing less damage and allowing it to be gently inserted into neurons to measure potential changes in a specific neuron. This technique, seen as quite ingenious and convenient in the 1940s, allowed scientists to easily measure any neuron they wished to study. Consequently, scientists quickly clarified the functional divisions of many brain regions and neurons. For example, by inserting an electrode into a neuron in a monkey and letting it play a game, if the ball entered the square and the neuron fired, it indicated that this neuron was related to the game. By inserting another electrode into a different neuron and letting the monkey play another game, if that neuron also fired, it meant that this neuron was related to the other game. With this technology, scientists' understanding of brain neurons advanced rapidly. The brain area responsible for vision works this way, and so does the brain area responsible for movement. When you want to make a movement, neurons in the brain also fire. However, motor control by brain neurons has an interesting characteristic that became key to the realization of brain-computer interfaces. Simply put, when a person makes a movement, there is a preparatory process in the basal ganglia. First, before you make the movement, the neurons in the brain related to this movement become excited. As you can see, our actions initiate through such a process, retrieving preparatory actions from the brain's action library, inhibiting disinhibition, and initiating the action. Therefore, different pathologies in the basal ganglia can cause two diametrically opposed diseases. One is where inhibition is too strong, leading to difficulty initiating movement, known as Parkinson's disease. The other is where inhibition is too weak, leading to involuntary movements, known as Huntington's disease. Since the relevant neurons in the brain are already preparing many things before we make a movement, this means that if we can detect the firing of these neurons, we can send these electrical signals out to drive external machines. Even without us making movements, we can control external machines purely through the firing of these neurons. Isn't this the brain-computer interface controlled by thoughts that we talked about? This idea is indeed quite good. So, scientists decided to take an innovative step, implant electrodes inside neurons, hoping to predict movements by monitoring the firing activity of a specific neuron and use machines to execute these commands. However, the results were not as perfect as expected. Trying to control machine operations through the firing of a single neuron proved to be quite unreliable. Simply put, the accuracy was greatly reduced. When you perform diverse movements, the monitored neuron will respond accordingly, making accurate interpretation extremely difficult. Now, suppose we detect that one of your neurons is firing. The question arises, what exactly do you want to do? The fundamental reason for this outcome is that our understanding of the brain is still at a very preliminary stage. Relying solely on the firing of a single electrode to predict a person's behavior is obviously unrealistic. On the other hand, we must recognize that the brain relies on a vast number of neurons working together to think or control any activity. The neuron you detect is just a very tiny, very limited part of the whole complex process. This is like trying to predict the entire weather condition by monitoring the most vigorously moving air molecule. The result is predictable and naturally unreliable. Since a single neuron cannot handle this task, scientists have to seek more advanced technological means, increasing the number of electrodes to improve the accuracy and reliability of monitoring. In the late 1980s, with the significant improvement of computer data analysis capabilities, many brain scientists began to turn their research focus to neuronal ensembles. In 1987, Miguel A. Nicolis, who was struggling with his doctoral thesis, happened to read a book about the Big Bang, the creation and evolution of the universe. A picture of a radio telescope in the book fascinated him deeply. This picture displayed the radiation distribution in space in a three-dimensional form, bringing him great inspiration. At that moment, an idea flashed in his mind: if we replace stars with neurons, radiation intensity with discharge intensity, and add a time dimension, can we record and scan brain activity? In the following years, Nicolis dedicated himself to the detection and research of neuronal ensembles. Their experimental equipment could already monitor the activities of 48 neurons simultaneously and record these activities over time. Through these data, they could form a three-dimensional image, looking like ripples on the water surface. Using these images, they could clearly observe how electrical waves spread among related neurons when a mouse's whiskers move. However, these data were far from sufficient. They knew that when a whisker moved forward, brain waves would produce such ripples; when it moved backward, it would produce other ripples. But how could they find patterns from these complex ripples to predict whisker movements? Smart folks may have already thought of it. Yes, here comes the power of artificial intelligence. Nikolilis's team decided to use artificial neural networks, ANNs, to analyze these data and find the internal connections and patterns between data and actions. In terms of data analysis and pattern discovery, artificial intelligence indeed demonstrated its powerful capabilities. Soon, the ANNs, through extensive training and learning, grasped the correspondence between data and actions, finding a close connection between neuronal discharges and movements. This meant that they could now predict mouse behavior to some extent by monitoring the discharge activities of these 48 neurons. Therefore, in 1997, they conducted a bold experiment, enabling mice to drink water with their minds through a brain-computer interface. The design of the experiment was as follows. First, they trained mice to use a lever to drink water from a cup. Then, they implanted electrodes in the mice's cerebral cortex, which could simultaneously record the activities of 48 neurons. These neurons were mainly responsible for premotor activities, i.e. activities related to movement preparation and execution. Next, they recorded the activities of these neurons when the mice used the lever to drink water and input these data into the ANNs for training and learning. After accumulating enough data, the ANNs were able to excellently recognize the discharge of these 48 neurons when the mice pressed the lever. Then, they disconnected the lever, meaning that pressing it would no longer have any physical response for the mice. This frustrated the mice, who tried everything to drink water. At this point, the researchers connected the drinking device to the electrodes. When the ANNs identified the premotor activities of the mice wanting to press the lever to drink water, they activated the device to bring water to the mice. In other words, when the mice wanted to drink water, they only needed to think about it, and the water would automatically be brought to them. The mice soon discovered their special ability. They could drink water just by thinking about it. Thus, they became the first mice in the universe to learn to drink water with their minds. However, there are huge differences between the brains of mice and humans. Therefore, Nikolilis's team next decided to shift the experimental subjects to monkeys. They further upgraded the experimental equipment to monitor data from 2,000 neurons simultaneously. Through another round of training and learning, the data successfully taught monkeys to use their minds to control a robotic arm to play games and drink rewarded juice. More importantly, with more monitoring data, they discovered some interesting phenomena. Some neurons began discharging during the motor preparation stage, the premotor stage mentioned earlier. These neurons are likely closely related to motor preparation activities. This discovery undoubtedly provided new clues and insights for a deeper understanding of how the brain works. There are also specific neurons that only discharge when controlling the robotic arm. This means that when the monkey realizes it can manipulate the robotic arm just by thinking, the brain mobilizes a group of specialized neurons to handle this task. This reveals an important fact. This completely external device has actually obtained a dedicated neuronal quota in the monkey's brain, being accepted and regarded as part of the body. This is the most crucial point. The success of our brain-computer interface technology does not lie in our cracking the mysteries of the brain or manipulating its coding to control external devices. Rather, it is because our brain demonstrates great inclusiveness and adaptability, accepting these external devices. The brain is so powerful that it constantly and dynamically adjusts the boundaries between you and the external world, defining what belongs to you and what does not. It is because of its inclusiveness and power that brain-computer interface technology can demonstrate unimaginable development potential. Perhaps you're still feeling a bit confused right now. Before delving deeper into further technological advancements, let's first discuss two experiments. The first experiment concerns phantom limbs. The medical community has long discovered that amputee patients often experience a terrifying phenomenon called phantom limbs, where they feel that the amputated body part still exists, accompanied by spasms and pain. Initially, the medical community was helpless, unable to explain the cause of this phenomenon or how to treat it. However, brain neuroscientists later found that it all stems from overactive neurons. The brain does not allow neurons to remain idle, so when an arm is amputated, the neurons that originally controlled this arm will take over the work of other neurons to avoid becoming unemployed. As these neurons, carrying painful memories, work in their new positions, they occasionally reactivate the connections that record pain due to normal discharging, causing patients to feel that their arm has returned, accompanied by unbearable pain. To eliminate this sensation of the amputated limb, doctors designed a treatment method called mirror therapy. Specifically, a box is placed in front of the patient, and they are asked to put their intact arm inside, with a mirror placed in the middle. Due to the reflective properties of the mirror, the patient seems to see their other hand. When the patient moves their intact hand, they feel as if the non-existent hand is also moving. In this way, their neurons gradually believe that the hand still exists and is no longer painful, gradually eliminating the painful experience. After a period of training, the sensation of the amputated limb will disappear. This phantom limb experiment illustrates 2 points. Firstly, your brain's functional areas are dynamically changing. If an organ disappears, the idle neurons will take over other tasks. This is why people who are blind have more acute other senses. Secondly, the boundaries of your body are defined by your brain. When a part of the body suddenly disappears and the brain has not adapted to this change, it will continue to perceive that part of the body as existing. Furthermore, using similar experimental methods, we can even get the brain to redefine the boundaries of your body, treating a fake arm as a real one. This is the famous rubber hand experiment. In this experiment, you sit at a table, place your left hand farther away and cover it with a board, and then put a fake hand in front of you. You stare at this fake hand while someone brushes your real and fake hands back and forth with a brush. After a while, you will feel that your real hand has moved to the position of the fake hand. At this point, if you close your eyes and are asked to touch your left hand, you are likely to reach out and touch the fake hand. What is the relationship between these experiments and the brain-computer interface, BCI, we are discussing? Why did we take such a long detour to introduce these 2 experiments? Because this is the key to understanding why BCI can succeed and what it can really do. Since the brain is constantly defining the boundaries of the self, the external devices we newly add can be accepted by the brain and truly become a part of you. Since the brain can always accommodate and allocate new neurons to external devices, our experiences can be expanded. With different peripherals, we can directly feel magnetic fields, see ultraviolet rays, hear ultrasonic waves, and may even directly receive and understand radio waves. Furthermore, if peripherals of your brain are everywhere in this world, your self can expand to the entire world. This is the ultimate vision of BCI, neither the downloading of memories and knowledge, nor the strengthening of the body, nor playing virtual games in the metaverse, but yourself ultimately escaping the prison of the body and becoming omnipresent, your experiences ultimately transcending the limitations of the senses and becoming omnipotent. You are everything in the world, and everything in the world is you. Such experiences and feelings may be beyond the imagination of our current group of humans who are still in the Neolithic era. Of course, this imagination is somewhat distant. But even for us in the Neolithic era, these characteristics of the brain are of great significance. This is the key to the amazing applications of our BCI, even in its extremely crude and primary stage. For example, controlling robotic legs on the other side of the Earth, becoming an electromagnetic human, and using your brain to control another person's body. Although these sound like fairy tales, Some animals on this planet have already experienced different things by participating in human experiments. These ideas have already been realized in experimental animals, meaning that from a purely technical perspective, there is no difficulty for humans. In the next issue, we will talk about the current status and challenges of BCI.